Meiosis 1 begins with prophase 1. In prophase 1, the chromosome condenses into egg-shaped structures that can be easily seen under microscope. Each chromosome consists of two sister chromatids containing identical genetic information joined by centromere. And this is centromere. These sister chromatids then exchange chromosomal material. This process is called crossing over. After crossing over, the sister chromatids are no longer identical to one another. This is one of the reasons why no siblings are genetically identical unless they're twins. Then the nuclear membrane disappears and the homologous pairs of chromosomes line up in the middle cell. The spindle fiber extends across the cell, and we have the next phase, which is the metaphase 1. I will draw the spindle fiber in color violet and connect the chromosome to centromere. And the metaphase 1 begins. The spindle fiber attached to one chromosome of each pair. The next phase is anaphase. During anaphase, phase 1, during this phase, the homologous chromosomes separate and start moving to opposite poles of the cell. This is the anaphase 1. So the final step is the telophase 1. In this phase, the spindle fiber starts to break down. The nuclear membrane reappears, and finally, the cell divides into two daughter cells after cytokinesis. That's the nuclear membrane. And we divide it into two. So the cytokinesis starts, cytokinesis is the division of cytoplasm. Telophase. These cells will now undergo meiosis 2 to another PMAT. In prophase 2, the chromosomes condense again in each of two daughter cells, and nuclear membrane disappears and the spindle fiber form. So the major difference between prophase 2 and prophase 1 is that the daughter cell only one copy of the homologous chromosome. So in prophase 2, there is no crossing over. That's the difference of prophase 1 and prophase 2. So now in metaphase 2, the chromosome line up again in the equator of the cell. The centrioles move to the poles of the cell. The spindle fiber attached to each sister chromatids. I will erase the letter or the cell division prophase 1, prophase 2 into a metaphase 2. So I will attach the centrioles of these sister chromatids into a spindle fiber. That attach to its sister chromatids. So we're going to the third part of the meiosis 2, which is the anaphase 2. In anaphase 2, the sister chromatids were pulled apart as the spindle fiber shortened. Also, the end of the cells are pulled further apart. The sister chromatids is pulled apart, and I thrown away the centromere. So the telophase to the nuclear to So in the telophase 2, after this anaphase 2, the nuclear membrane will reform cytokinesis. In telophase 1, the telophase reform cytokinesis occurs and the cells are officially divided into 4. So that's the nuclear membrane. Touch again in the chromatids, and then they are divided into 4. Since meiosis 2 began with 2 cells and each of those cells further split into 2, we now have the 4 unique granddaughter cells at the end of the meiosis 2. Mitotic cell division. During the first stage of mitosis, that is prophase, the chromosome become visible and can be seen clearly under the microscope and the nucleolus and the nuclear membrane disappear. The spindle fiber elongate to attach with each chromatid at the centromere. So the next comes is metaphase. During this phase, the sister chromatids align at the equator or the center of the cell. So both chromatids face the opposite ends of the cell. Now the sister chromatids are ready to be separated. And the spindle fiber is still attached to the centromere. The spindle fibers will help separate the sister chromatids during the third phase. The next phase is anaphase. During the third phase, the spindle fiber is now shortened and the sister chromatids separate. Additionally, the poles of the cell move to further apart, causing the sister chromatids pull apart. So in the telophase is the phase telophase is the phase where the components of the new cells begins to appear. At this point, the spindle fibers are broken up and the new nuclear membrane surrounds the chromosomes. The chromosomes uncoil and return to an uncondensed state. After all the phases of mitosis, the cell undergoes cytokinesis, which divides the cell into two halves. The cell organelles are replicated by now. They are now split between the two halves of the cell. The two daughter cells produced as a result of mitosis are identical. Now, the purpose of the mitosis is to help us to grow and also to help us in our wounds to heal. And I guess this video will end here, and thank you for watching.